We are.
Okay. All right. So I'll share my screen again. So good morning, everybody. My name is Arti Ahluwalia, and I'm a professor of biomedical engineering at the University of Pisa. Can you see my shared screen now?
Yes.
Okay, so professor of biomedical engineering at the University of Pisa, and I'm also director of the research center ePiaggio, which is a research center in robotics and biomedical engineering at the University of Pisa, member of the Department of Information Engineering, and also a member of the Italian Inter-University Center for the promotion of the principles of 3 R's in teaching and research. Now, if you don't know what the 3 R's are, you're going to learn about them in my presentation. And I'm going to talk to you about advanced biomedical tools for modeling biological systems, with particular reference to something that's very relevant at the moment, which is modeling of the lung and the interaction of the lung with nanomaterials. So the study of the lung and modeling the human lungs has been an ongoing topic of research for a number of years. And it's a very, very complex system. As you can see, we have a complicated airway system whereby we inhale air and particles in the air. They go down through the upper respiratory tract and then through the lowest tract where we have the bronchi and the alveoli. And at the level of the alveoli is where we have the exchange of materials and gases which are in the air and go into the bloodstream. So that's where the actual exposure of substances occurs, at the level of the alveoli and a little bit in the bronchi, but mostly in the alveoli. And modeling the human lungs is very, very important, not just for a basic understanding of how we breathe and what happens to substances that we inhale, but also for a lot of diseases, for instance, viral infections, which are transmitted through the airways, the effect of smoke on our lungs, so nicotine, tobacco, for the development of drugs, for example, for asthma, for studying lung cancer, the effect of sports, etc. So there are many, many different applications, and it's a very, very complex system. So we use a lot of different types of models to try and understand how the lungs work and how they get ill. And if we look at models in biomedical engineering, we can define a model. Basically, a model is a representation of reality. It's something that we use to try and understand what's happening in the real world, to try and predict what happens. And there are different types of models. For example, we can talk about mathematical models. When we talk about biomedical engineering models, usually we refer to in silico models, because quite often these mathematical models are done using a computer, so they're called in silico. Or very, very often in biomedical engineering and in biomedicine, we use animal models. So we use an animal to represent what happens in the human body. And these are known as in vivo models. And then finally, in biomedical engineering terms, in biomedical terms, we can also make a model in the lab. For example, we can make a small model of the lung, or we can make a real tangible model of a human patient, which many doctors use or student doctors use to do their to practice. And these are known in biomedical terms for development of disease models. They're known as in vitro models. So different types of mathematical models have been developed. For example, there are many models of patients and people as components of a multi-compartment system. Animal models are very, very common in the medical field. So for instance, here you can see these are rats being exposed to smoke to see what the effect of smoke is on their lungs. And then finally, the lab models, many different types of models here as well. In biomedicine and biology, we use in vitro models, which are made up of cell culture systems. And so all these are used for the development of different aspects of the development of models of different aspects of the human body. So just comparing in vitro and in silico models with the animal models, the animal models are very, very heavily used in biomedical science. And compared with in vitro and in silico models, obviously you can represent the complexity of an entire organism, which is something that you cannot do in a simple computational model or in a cell culture dish. Whereas in animal models or in vivo models obviously have problems too. For example, they have ethical problems. It's not very nice to use an animal and maybe hurt it or kill it in order to develop our knowledge on different aspects of the human body. And then there are clearly differences between humans and mice or humans and dogs related to their metabolism in terms of the structure of the gas airways. There's a very different architecture as well. And then animal models are usually very, very expensive because you need to maintain animal housing, you need to feed the animals, you need to make sure that they're all in good conditions to be able to carry out your experiments. Nevertheless, animal models or in vivo models are, I would say, the principal way that we develop our knowledge in science at the moment. For instance, even at the regulatory level, it's important to use animals to test substances that can be potentially inhaled, so substances that are used in industry, substances that are used on a commercial scale, even the manufacture and selling of vehicles and so on and so forth. We have to test the fumes on animals. And animals, there is actually regulatory tests that you have to do. So animals are exposed 5 days a week, or even 7 days, so the whole week. And then you have to use males and females as well to see if there are sex differences. So these are tests that are required before you can commercialize a substance. Obviously, in vivo, or rather animal models, are important for the development of vaccines. It's very important when you develop a vaccine, for example, even for COVID. We must go through phases of animal testing to ensure that the vaccines are safe and don't cause harm. So here we go through phases of what's known as preclinical testing before applying vaccines to humans. And then a huge amount of research goes on in diseases. So for instance, to understand about chronic obstructive pulmonary disease, animal models are quite often used. So we try and provoke these diseases in animals to see what the effects are. And as you can see in this review article, we use mice, guinea pigs, rats, and other animals. So as I mentioned, there are, however, major differences, particularly, well, almost in every organ, but particularly in the human airways, which is the focus of my talk. So the main animal that we use to simulate or model the human lungs is the rodent, so the rat or the mouse. more often the rat than the mouse because it's a little bit bigger. But there are huge differences between our airways, both in terms of the architecture, as well as in terms of the metabolism and cell composition. So one of the main differences that between the lungs of a human and a rat are that rats only breathe through their noses, so they don't breathe through their mouths, whereas humans breathe quite a lot through their mouths, especially when we're talking or doing sport. So that's a major difference. And then there's also a large difference in the way that the branches deviate from the mother branch. So you can see here the human airways have have two major branches which then branch out into smaller bronchi and alveoli, whereas the rodents have one major trunk and then all the bronchi and alveoli branch out from that. So there are a lot of differences between the two, and you can't really compare and predict what happens in a human from what you observe in an animal. And not only because of that, but because of ethical motivations, because a lot of people feel that it's wrong to use animals in research. It's wrong because animals will suffer when they're exposed, for example, to vaccines or given harmful fumes. For example, they're forced to inhale cigarette smoke. And because of this ethical problem, way back in 1959, so well over 50 years ago, two English scientists called Russell and Birch, respectively, wrote a book on how to reduce the suffering of animals in experimental science, and what type of principles to apply when you're doing experiments on animals. And these principles are known as the 3 R's. So the 3 R's are basically principles that we have to use when we do animal experiments. So the first thing we have to make sure is that there are no other methods that don't make use of animals, and this is known as the principle of replacement. So we want to try as much as possible to replace animal studies with methods that don't use animals. And then if we can't replace them, we must try and reduce them. So we must try and use as few animals as possible to get the best possible results. So try and design an experiment in which you minimize the number of animals used. And then finally, if you have to do an experiment with an animal, it's important to refine your techniques, make sure that you use anesthetics, make sure that the animals are well looked after and not stressed. And so these are known as the 3 R's. And in Europe, they've actually been formally adopted as legislation in all EU countries through a directive that was passed in 2010. So this directive is a directive on the protection of animals for scientific purposes, and it applies the 3 R's principles. And that's basically what underlines most of the research that I do and that my group does. Now, going to the specifics of research on the lung and the way that particles enter into the lung system. Basically, we have particles in the air which enter into the lung when we breathe in. And these particles can interact with the lining of our lungs and cause inflammation and stress. And then that inflammation and stress, and also the passage of airborne particles into our lungs, then causes diseases. So this is a process by which we end up getting ill from things that enter into our airways. And in the case of the coronavirus particle, which has a typical diameter of 120 nanometers, so it's actually a nanoparticle because it's on the scale of less than a micron. These particles enter into our lungs in the form of droplets and aerosols, And we have to try and understand how they enter into the lungs and how they deposit onto our alveoli, and then how they translocate and enter into the bloodstream. So, you know, the processes that we want to study are what happens when somebody breathes out, when somebody exhales, and whether there's a difference between a sneeze and a cough, or just simply breathing and talking, and how this then translates into infection into the lungs. So here, a lot of work has been done on studying how airborne particles and aerosols disperse through the air and then get transmitted into a person's lungs and then into his bloodstreams. And a lot of these studies are quite interesting. You know, they show how a sneeze travels much further than a cough, and of course travels much further than simply breathing, which is pretty obvious. But you know, it's very interesting to see that sneezing will actually almost increase in distance by almost 10 times with respect to just breathing, and particularly breathing with your mouth closed. The other aspect that we need to study then is how these particles, be it viruses or pollutants or even an inhaler like for asthma, how these particles enter into the lungs, and how the movement of the lungs actually conditions the way that the particles finally sediment onto the alveoli and move across the lung barrier. Because it seems that the movement of the lungs is a very, very important vehicle for the entry of particles into the bloodstream. So these are all aspects that need to be modeled, and which one would seek to study in vivo, in vitro, or in silico models. And I'm going to talk to you about models that don't use animals, because as I mentioned to you, we're guided very much in my research by the principles of 3 R's. So just to look at how particles move with breath, because it's a topic of interest at the moment, how they move with breath, coughing or sneezing. It's very important to understand that most of the particles that come out of our breath are in the nanoscale. So basically the scale of viruses. So when we breathe in or out, we very easily inhale nanoparticulate matter. It's much frequently generated from our breath than microparticulate matter. So you can think that whenever we breathe out, or we sneeze, or we speak, and we have particulate matter in our lungs, for example, viruses, these come out in the form of little droplets, which basically have one coronavirus particle per breath, per droplet that we emanate, if we do have them. Or depending on obviously the amount, the infection level we have, it could be a lot less than one. But here you can see that you know, we do emit quite a few particles per centimeter cube. So from about 3 per centimeter cube to about 1 per 10 to 100 centimeters cubed. So this is what is actually being transmitted during this period of infection with COVID and infection of the virus SARS-CoV-2. Sorry. Important then is to use models of aerosol transmission and formation, and see how these aerosols move through the air, and how then they sediment onto surfaces, because we know that it's important to remember that even surfaces can contain traces of viral particles, and more importantly, how they actually transmit into the air. Because they're so small and so light, they actually travel quite a long way in the air, and will only sediment after traveling quite a distance, depending on the way that they're moving and the force with which they're emanated from someone's breath. So there's a lot of work going on in modeling the movement of nanoparticles, and here we make use of what we know in cloud science, because clouds form in the same way. So they form in droplets and then they condense and sediment. Now, going into the detail of what we're actually doing, well, what we're trying to study is to study how nanoparticles, be it viruses or smoke particles or even engineered nanomaterials, how they actually enter into the airways, how they deposit onto the alveoli, and then how they move across the barrier between the air and the bloodstream. So here on the diagram on the right, the air side is the alveolus and the blood side is the capillary. And what we're trying to do then is to create a model of this alveolar system during its movement. And I'm going to talk to you about how we developed our alveolar system and what we're trying to study at the moment, and how we have used engineering principles to make a model of the lung. So the typical diameter of an alveolus is around 500 microns. And normally, when one studies or develops lung models, we tend to use cell cultures of lung cells, so lung epithelial cells. Quite often they're cultivated in Petri dishes, but we now have much more sophisticated models that are based on what's known as air-liquid interface cultures. So air-liquid interface, we use the acronym ALI, so ALI cultures. And here what you can do is you can use a specialized cell culture dish, which is known as a transwell plate. And it's formed of a double layer. Here on the right you can see, or even on the left in figure A, you can see this double layer of compartments, and the cells are cultivated on a porous membrane. So it's like just a polycarbonate membrane with holes on it, and you put your cell culture medium on the top and on the bottom. And so if you have your cells on top of the membrane, you can put any material you want on the top and see how it moves across the cell layer into the bottom. And the very same thing can be done also at the air-liquid interface. So instead of putting a liquid on top of your cells, you just leave air. And so here you simulate the alveolus blood barrier. So on one side you have air, on the other side you have liquids, and you can put a substance here, in a smoke particle or a viral particle, and you can see how it is how it moves through the cell layer. And the cells are usually composed of an epithelial lining of the lungs when you're studying a lung in vitro model. There are also more complicated models like organoids and spheroids and lung on a chip. And I will show you how we developed a lung on a plate with these components and also other components that are present in the lung. So in fact, when we look at what components are present in the lung, we try and break down the system that we want to replicate to see what are the main features of the human lung. And then we try to model these as best as possible in our in vitro setup or in our laboratory. So as an engineer, we looked at the key elements of the alveolar interface, and we know what are the important aspects. Well, the most important thing is we have a cell layer. So these are the cells that line the alveoli. We know that the alveoli are moving, so we have a stretching and we have this continuous movement at around 0.2 hertz, so one breath every 10 or so seconds. The strain level, so the amount of stretch, is about 5 to 12%, but it can get to around 20% if we do forced breathing, if we have some kind of lung disease and we can't get enough oxygen in, the stretching levels are much higher. We have aerosol exposure, because that's how particulate matter enters into the lung. It enters into the lung and it becomes an aerosol because it mixes in with the very humid environment of our alveoli. We have an air-liquid interface, obviously, so we've got air on one side and then liquid on the blood side. And then we have the blood side is actually a moving liquid because it's movement of the bloodstream. And we know that the shear stress is of the order of 1.5 Pascal. So these are the engineering elements that we want to build into our alveolar interface. And if you go and look in the literature, there are many, many papers on lung in vitro models. There are systems with air-liquid interface and smoke exposure. You have smoking machines whereby you can actually put a cigarette on top of your cell culture and mimic smoke exposure. And there are several models with stretching as well, so you can actually stretch your membrane where the cells are. But none of these three aspects, so stretch, air-liquid interface, and fluid flow, are combined together, and that's where we saw a gap. And so we decided that that's what we would like to bring in. So we'd like to bring in all these important aspects into our lung model. And what we wanted to do, therefore, was to design a really physiologically relevant, so closely simulating the physiology of the human lung in an in vitro model. And an in vitro model that is then we could then use to better understand how particulate matter interacts with the lung, and particularly particulates of the order of nanometers, so the size of a viral particle. And that will enable us to get more predictive results of lung particulate interaction using the principle of the 3 R's. So what we did was we developed a moving air-liquid interface system. So it's not ALI or ALI, it's MALI for moving. So the system that we have developed, and I'm going to describe to you briefly, is an air-liquid interface bioreactor, basically, with a biological element, but in which we also have a moving lung membrane with cells on it, so with lung cells and a moving membrane that moves up and down, very similar to the way that the lungs move when you breathe. And also to which we can expose aerosol at the same time, so we can study the way that the aerosol interacts with a moving interface. And what we also added was the flow of the bloodstream in the bottom chamber where we simulate the blood flow. So we have here a very, very simple representation of the system. It's basically 2 chambers, a top chamber, which is the apical chamber, a bottom chamber, which is the basolateral chamber. They're separated by a membrane, which is porous, and the lung cells are cultivated on top of the membrane. The bottom chamber has the flow of liquid, which represents the bloodstream, and the top chamber has an aerosol dispenser, so basically a generator of clouds known as a nebulizer. And the whole system is regulated and controlled so that we can monitor the movement of the membrane, we can monitor the way the cells behave, and we can also change the stretch and the frequency of movement of the membrane. So just to briefly show you what the system consists of, it consists of the key elements that we identified in as requirements for a sort of physiologically relevant lung model. So movement of the membrane, the aerosol, the shear stress on the blood side, and also the capacity to monitor what happens in real time. And that's what we call physiologically relevant because it moves closer and closer, even in complexity, to the actual human situation, which enables us to get more predictive and more meaningful results, while at the same time reducing the number, or well, in this case, replacing hopefully the animals used in testing. So what we did first was we developed in silico models. So we looked at how particulate matter sediments onto the membrane, and whether this sedimentation is similar to what happens in the alveoli. So we basically made a model of an alveolus, and that's basically what our MAI system represents. And as you can see here, it's upscaled by a factor of 100, but that's okay. It enables us to see it better. So the system was actually scaled so that the membrane represents an upscaled version of the alveolus, and the input where the nebulizer is and where the droplets form is scaled to the size of the alveolar tracts. We developed, therefore, computational models, and here, if you want to look at particles moving through a system, you've got to use Eulerian models, so you can't use the Lagrange systems because you're looking at individual particulate matter. And we studied the way in which particles are formed and how they deposit onto the surface of the membrane as it moves. And here you can see the particulate matter moving down as it's nebulized at the same frequency as we breathe. So basically we were creating a cloud, or rather a group of particles every couple of seconds to imitate the frequency of breathing. And we can see here how the particles deposit onto the membrane. So this is just a picture of a picture of how the particles sediment during each breath. And what we see is that every time we breathe in, there's a complete coverage of an alveoli in about 30 seconds if we're in the presence of a continuous cloud of substances coming in. We also developed analytical models of the moving membrane as we push the membrane down by increasing the air pressure, as happens in the lung. And so what we do is we overpressure the apical chamber, the top chamber, and so the membrane will move up. And then when we exhale, so we underpressure the chamber, the membrane moves down when we overpressure the chamber and up when we take the pressure out. And so that simulates the breathing motion. And we therefore developed models of the basal chamber using here Lagrangian dynamics. So looking at the way the membrane will move every time we increase the overpressure in the top chamber. And here you can see the model that we developed using fluid structure interaction and laminar flow regime on the bottom blood side. And these are the parameters that we used. And then what we had to do was we had to develop a control system to cyclically increase and decrease the pressure in the top chamber. So we had to use pressure regulators and electrovalves that turned on and off every at 0.2 hertz. And obviously we were able to change the pressure as well according to the amount of force one uses to inhale. And then at the bottom on the blood side, we have a peristaltic pump, and that too is controlled in its flow rate to be able to change the shear stress on the bottom side. So we have this bioreactor, we have a membrane, which is very, very flexible. It moves up and down at a frequency of 0.2 hertz. We can monitor the level of stretching of the membrane using a optical system, or even looking at the changes in water level in the entire fluidic circuit. And then obviously there's an aerosol generator on top of the apical chamber, which releases a cloud of nanoparticles every time the system breathes, so every time the system moves down. And there's a control box that controls the movement and the timing. So here you can see the prototype. You can see the membrane moving at the bottom there, and this is the Mali bioreactor during its operation. So we can actually quite nicely replicate the movement of the lungs. We can quite nicely replicate the strain and the frequencies, as well as the pressures, and obviously the deposition of nanoparticles, be they viruses or smoke or pollutants, or whatever one wishes to study. In the particular case that we are analyzing, we're looking at generic nanoparticles. So for the moment, it could be any type of nanomaterial. We're looking at the impact of different sizes on the different sizes of particles on the system. The aerosol generator is actually a very, very simple commercial device. So this is actually the device that's used to generate aerosol for therapeutic purposes. So if you have a problem like asthma or something, you use these nebulizers that pump medicine into your lungs, and you can regulate the frequency and the duty cycle of these systems quite easily. So this is what we used, and this was just placed on top of our apical chamber. The system is very, very simple to use. You've got a little top, and you can fill it with any type of nanomaterial that you want, and it will nebulize at a frequency and on-off cycle that you can quite easily control. In fact, the control box controls everything. It controls the actuation of the membrane, it controls the aerosol deposition, and it also controls the flow of fluid in the liquid compartment. So just looking at it, it's a very simple system, 2 chambers and a stretching membrane, but obviously the complexity lies in the design to make sure that we get a uniform particle deposition, and to make sure that the stretching level of the membrane is very similar to the stretching that we get when we breathe. In fact, that's a critical component of our system, because we need to have a membrane that is very, very elastic, very, very flexible, doesn't have any delay or any hysteresis, and is also porous, because we want to see how particles will go through the pores and enter into the bloodstream. And it has to be very, very biocompatible, because we want to seed cells on it, and we want to make sure that the cells function and behave like the human lung barrier. So a lot of effort was made to select a good membrane, and in fact, I have to say it's one of the critical points of the system, the membrane. So we're still trying to find the ideal membrane. So we have studied different materials, and we're still looking at developing better materials. So the material that we used in the experiments that we published is a polycarbonate urethane elastomer. It's very, very flexible, extremely durable, and it has a pore diameter of 4.4 to 5 microns, a thickness of 70 microns. And importantly, it's highly elastic and stays elastic throughout experiments that last for up to a week. The cells, we seeded lung epithelial cells on the membrane, and here you can see a confocal stack. The cells will nicely penetrate into the fibers, and they'll colonize the membrane, and therefore we form a very compact 3-dimensional cell layer, which in many ways mimics the alveolar barrier. So we're quite happy with these results so far. You can see that the cells are quite well organized in the system. So in conclusion, I just wanted to show you briefly how you can develop complex models of the human body, in this particular case of the human lung system, using in vitro techniques. So the important thing that you have to do is to look at your organ or your system that you want to replicate. Look at it as an engineer and look at the different components. So you have to do a kind of a re-engineering of your organ or your system. So that's exactly what we did. We looked at the lung and we said, what are the important components for an engineer? So, you know, we have movement, we have pressure, we have flow, etc. You have to try and quantify these and then try and replicate them in an upscale or a downscale system, and then use this to construct a complex three-dimensional system. So this is exactly what we did. This is how we started off. We started off as, you know, just thinking what's the design principle behind the lung, and we developed this physiologically relevant in vitro model of the alveolar interface using the principle of the 3 R's. So, you know, we wanted to use something that didn't make use of animals. So that's why it's known as an in vitro model. But as you can see, very much supported by in silico and mathematical and finite element models. We can say that the system replicates the key elements of the alveolar interface. And so we can use it, and we are using it, to study the interaction between airborne viral particles and the breathing lungs. So this is what we intend to do now that we realize that the SARS-CoV-2 is basically a nanoparticle and interacts with our lungs as many nanoparticles do, and is transported in our lungs as many nanoparticles are. And so we can use this for different applications, in particular in the current pandemic. We could use it to test the efficacy of different vaccines, use cells from patients and for people who have also recovered to see what the differences are in response of these two cell types, and then therefore study how and why some people are resistant and some people are more susceptible to infection. And we do think, and there's literature to prove or to suggest, that it's actually the stretching of the lung that increases the probability of entry of viral particles into the bloodstream. So the stretching process is very, very important in the study of viral particle-lung interaction. So just to finish off, I'd like to say thank you to all the people in my lab, in particular to Daniele Cei, who developed much of this work, Roberta Nossa, who took up a lot of it for further studies, and the rest of the group, as well as the lung comprehensive pneumology center in Munich in Germany, with whom we conducted a lot of this research. So thank you very much for your attention, and I'm happy to answer any questions.
So thank you for the talk. Question?
Thank you, Andres. I have to say that the images are thank you to Roberta, because they were part of her PhD presentation.
So let me stop the recording.